I think, David, you can begin.
Good afternoon. I'm David Herman. I'm a neurologist at the University of Rochester, involved in the field of CMT research and with an interest in preparing for clinical trials that hopefully will be plentiful in the years to come. By way of introducing this topic today, the Inherited Neuropathies Consortium, or INC, is a consortium that was developed about 15 years ago and funded by the NIH Rare Disease Clinical Research Network to prepare the field for conduct of clinical trials. And the INC has focused on a number of areas. We've been involved in defining the natural history or progression of different forms of CMT. We have developed different patient-reported outcome measures, physical functioning assessments, and different biomarkers for use in trials in CMT. And importantly, the INC has been integral to the discovery of new genetic causes of inherited neuropathies. And discovery of new genes for CMTs has really created a window of opportunity for treatment. For example, just a couple of years ago, the genetic cause underlying a recessive form of CMT was found to be SORD deficiency, and this has rapidly led to the initiation of a clinical trial. The initial work of the Inherited Neuropathies Consortium utilized a simple clinical assessment known as the CMT examination score to understand the natural progression of CMT and different forms of CMT ranging from CMT1A to CMT4C. And what we've learned is that most forms of CMT, aside from being quite rare, are also very slowly progressive, with progression detectable on their CMT exam score over years rather than months. And this presents unique challenges in trying to determine whether treatments are effective in clinical trials. Because ideally, clinical trials need to be conducted over 1 to 2 year timeframes, and thus detecting treatment effects when diseases are very slowly progressive, such as CMT, poses quite a challenge. To this end, the INC has worked on developing additional outcome assessments and biomarkers to potentially more sensitively detect treatment effects in clinical trials. And thus today we have a validated infant scale for use in clinical trials in CMT. We have a validated CMT pediatric scale and a validated quality of life measure for use in childhood clinical trials in CMT. We have parallel measures for adult clinical trials, including the CMT functional outcome measure and the patient-reported CMT health index. The INC has also developed and is validating a range of biomarkers for use in clinical trials in CMT. We're assessing wearable sensors to measure physical activity and gait and balance in different forms of CMT. Much work has been done using imaging to assess disease progression in CMT. So measuring the fat content of leg muscles in MRI in the lower limb is a very sensitive way to pick up change and progression of CMT over a 12-month period. We've initiated studies to assess sensory nerve endings in the skin non-invasively with a technique called confocal microscopy of Meissner corpuscles. There's also been work done on skin biopsies to provide a window into the biology of different forms of CMT, and it is hoped that skin biopsies can be used in clinical trials to assess target engagement, to determine whether prospective treatments are having the desired effect on the biology of different forms of CMT. And finally, study of blood samples has provided a rich source to identify potential biomarkers or disease markers, which hopefully will give early indicators of response to treatment in different clinical trials. And so today we have a range of specific validated functional outcome assessments, patient-reported outcome assessments, and different biomarkers that can be used for clinical trial design in CMT. And the challenge to the field, as we now have a rich pipeline that we'll hear about today of potential candidate therapies, is how best to use the different outcome assessments and biomarkers. Which outcome assessments and biomarkers should be used for which subtypes of CMT at trials at different stages of disease, which are the appropriate outcome measures and biomarkers for early versus later intervention, and what are the appropriate assessments for different types of interventions? What assessment should be used in gene therapy trials versus small molecule trials? Finally, there's much work needed to be done for the CMT expert community to engage with regulatory agencies. We need to gain a common acceptance and understanding of what the optimal trial design looks like for CMT trials and how to measure effects of treatments in future trials. And with that, I'll turn the discussion over to Dr. Dubensky.
Thank you, David. So, hello, everyone. Good afternoon. This is Tom Dubensky. I'm a member of the CMTA on its board and someone who lives with CMT1A. I hosted and moderated this panel last year. So like everyone else that's on this call, I'm anxiously anticipating the discussion today and seeing the progress that's been made over the last year. So let's start by introducing our panelists, each of whom have significant positions in the biotech companies that we will be hearing about today. The first is Dr. David Solomon, who is the CEO of Pharnext Therapeutics, which is a French company. The second panelist is Dr. Suyash Prasad. Suyash was with us last year. Welcome back. And he is the Chief Medical Officer of Taysha Gene Therapeutics, which is located in Texas. And then our final speaker is Dr. Shoshana Shendelman, who is the CEO and founder of Applied Therapeutics. So I thought the way we would approach this, and Shoshana, first of all, was with on this panel last year, and it's nice to see both you and Suyash again, and thanks so much to both of you and you as well, David, for participating today. What's exciting to me is that two of our panelists are engaged in advanced clinical studies, and so we're very excited to hear about that. Excuse me, that's Dr. Solomon and Dr. Shendelman, and both of whom are developing therapies for CMT, and I'll let them talk about their companies. And then Dr. Prasad works at Taysha, as I said, as its CMO, and Taysha is a gene therapy company, which is, as all of you on this audience are well aware, is a therapy that's gaining so much steam, and also has curative intent. So it's really very exciting. So let's start with Dr. Solomon. He is the CEO of Pharnext Therapeutics. They are in late-stage development with a drug known as PXT3003. And so let's start, maybe, Dr. Solomon, by talking to the audience about this drug, its mechanism of action, and if you can say a little bit about its stage of clinical development and what you're observing in the clinical studies that you can talk about.
Yes, thank you so much for your introduction, and of course, a pleasure to be here with all of you and with everyone viewing. Our company, Pharnext Therapeutics, is in late-stage clinical development for a medicine, as Tom said, called PXT3003 for CMT1A. This medicine is a fixed-dose combination of 3 existing medicines, and the medicine is in late-stage clinical trials. We're conducting, under FDA sort of guidance, a pivotal phase 3 study called Premiere that you can see at clinicaltrials.gov. This is a study that has already enrolled 387 patients at 53 centers globally. As the study is fully enrolled, we expect the readout of this trial in CMT1A to be in the fourth quarter of 2023, so just a bit more than 1 year from now. This is a study that has enrolled patients according to the ONLS scale that have mild to moderate disease. The patients are measured using the ONLS scale, which is the primary endpoint of the study. And the patients are taking our medicine, again, a fixed dose combination of an oral medicine 3 times a day. The mechanism of action of this medicine is to lower the expression of PMP22. And as all of you know, in CMT1A, PMP22 is overexpressed or duplicated. And by lowering the expression of PMP22, we believe we bring back a lot of the features of normal neuronal transmission to patients. To wit, we've already had 5 years of extension study data, the same medicine, the same high dose, the same patient population globally. And we're following 123 patients today that continue after 5 years to improve according to ONLS on the scale. So we're excited for the progress to date, and we're excited for the readout that's coming. And we think that this is a promising avenue. One thing to mention, another biotech company in the US called Amylyx Therapeutics just had a fixed dose combination of a dual acting medicine for motor neuron disease, or ALS, approved yesterday. And so this idea of fixed dose combinations of existing medicines to treat rare neurologic disorders is back in favor, also with the FDA. So we press on. We are delighted to be part of this community. Our goal is to help patients and their families and their caregivers. And we look forward to any discussion you may have about our medicine.
Thank you, David. That's really very exciting, and as you indicated, and with completion of the Premiere study, I saw that the company just announced that that you have opened a open label extension study, and you've already enrolled a patient who has been on PXT3003, as you call it, for 5 years already. So that seems that it must be really an encouraging therapy. Obviously, patients wouldn't elect to continue to take this drug were it not tolerable, as well as encouraging in terms of their symptoms. And just wondering if you can comment on that.
Yeah, I mean, it's anecdotal, but the fact that 123 patients globally continue to travel many hours to see their physicians, be measured by ONLS and receive the medicine, and not drop out after 5 years suggests to us, again, anecdotally, that the medicine must have some effect, or else they would just drop out from the study. So that is encouraging, but of course, the results from a pivotal phase 3 study will be more definitive. We're working very closely with the FDA. We've done a SPA, special protocol assessment, to essentially work with the FDA to agree on exactly the protocol that they'd like to see for an approval, and we're crossing all the T's and dotting the I's as we speak. So again, the readout is Q4 23, and we believe this has terrific promise for the community.
Well, thank you, Dr. Solomon. I will speak for all of us that have CMT1A. We anxiously anticipate, that readout, and I can't wait for the day that we can possibly go see our physician and be prescribed that medicine. So with that, let's move on to the next person that is the founder, again, of Applied Therapeutics, and that's Dr. Shoshana Shendelman. Is working on a different type of CMT, but also is an advanced development in a what's on the website for the company is called a pivotal clinical study using a different approach, and also targeting CMT, so a different CMT2, a different subtype of CMT. So, Dr. Shendelman, would you please tell our audience about the aldose reductase drug that you're developing, and what its mechanism of action is, and if you can give us an update on your trial, please.
Sure, absolutely. Thanks, Tom. So we're developing a drug called AT007, for a specific subtype of CMT that David Herman referred to a few minutes ago as SORD deficiency. So this is a really good example of how the academic physicians and the Inherited Neuropathy Consortium and the patient organizations like the CMTA have been able to work together to identify the genetic cause of a subtype of about 10% of CMT2 patients who previously were categorized as having CMT2 based on symptoms, but they were sort of put in the bucket of genetic cause unknown. And it was because of the efforts of many researchers working together that they were able to identify the genetic cause of the disease, which is mutations in the gene coding for an enzyme called sorbitol dehydrogenase, or SORD. And this was also a really amazing discovery because we know what that enzyme does. And there's actually been decades of research into the pathway that this enzyme participates in. So sorbitol dehydrogenase, or SORD, is an enzyme that converts sorbitol to fructose for energy production. And it's part of an enzymatic pathway called the polyol pathway. It's a 2-enzyme pathway. The first enzyme is aldose reductase that converts glucose to sorbitol. And the second enzyme is sorbitol dehydrogenase that then takes sorbitol and converts it to fructose for energy. Patients who are missing this enzyme or have mutations in SORD are unable to metabolize sorbitol. So when aldose reductase converts glucose to sorbitol, it has nowhere to go. And sorbitol builds up to very high levels. And this results in neuronal degeneration and neuronal dysfunction. And the role of sorbitol in neuronal degeneration had actually been studied for decades in the context of diabetic peripheral neuropathy. This pathway is very well understood. We were already developing a CNS penetrant aldose reductase inhibitor for another metabolic disease. And so this was an opportunity to move very quickly. So this genetic identification of SORD deficiency was the cover of a Nature publication in 2020. So here we are 2 years later, and we're in a phase 3 study in SORD deficiency. So our drug, AT007, blocks aldose reductase, so the enzyme that comes before sorbitol dehydrogenase. So it's blocks conversion of glucose to sorbitol, so that even though sorbitol still has nowhere to go in these patients, it doesn't accumulate because you're blocking formation of it from the start. And this is what we call substrate reduction. And this is a funny pathway because it's actually not a primary metabolic pathway. You don't need to use this pathway for anything. You have other ways to metabolize glucose. So by blocking aldose reductase, you stop sorbitol production. And in patients who are missing sorbitol dehydrogenase, it stops, hopefully, this neuronal degeneration from happening. So it's a great example of academic research and the patient organizations working together to identify genetic causes of the disease that then provides a clear pathway forward for us for drug development. And so we're in a phase 3 trial right now. AT007 again is a CNS penetrant aldose reductase inhibitor, but it's an oral medication, so it's a liquid suspension that's taken once daily. We did a pilot study last year, and I think that was probably the update we had for last year's conference, which was in a small number of patients with SORD deficiency who took the drug in an open label format. We showed a very substantial reduction of sorbitol levels in their blood and tissues in a pretty short period of time. So it was rapid. You could see about a 66% reduction in sorbitol levels within 7 days. It was, sustained over the 1 month of treatment. But that was just a pilot study to see if this concept worked, if the drug really reduced sorbitol levels the way we thought it should. And so based on that, similar to David's comments, we then went and talked to the FDA about what does a pivotal or registrational study look like. And so our study is just in the final stages of recruiting right now. So we're almost fully enrolled. Hopefully, very soon we'll be able to announce full enrollment. And it's a 2-year study where we're looking at treatment of patients with AT-007 versus placebo. And we're looking at a few things in this trial. So we're looking at sorbitol reduction at an early time point, so at 3 months of treatment. We're also looking at muscle MRI, which David Herman referred to earlier. It's a sensitive measure of looking at muscle degeneration through fat fraction. And we're also looking at functional outcomes, things like 10-meter walk run, stair climb, sit to stand, and other components of the tools that David also was referring to. So CMT FOM, CMT high. So it really, I think, is a great example of why, understanding patients and their journey and their clinical stages is important. Developing tools to measure those effectively, because they are important for clinical trials. And so we're pretty hopeful, but we're still discussing with the FDA whether sorbitol reduction can be used as a biomarker for accelerated approval. We've powered the study for clinical outcomes, though, so there is a path forward for us if we do need to look at the clinical outcomes for approval. And it's all being studied in the same trial, which is placebo-controlled. So we're very excited about what this next stage is. And we're also very appreciative to the researchers that allowed us to move so quickly to this phase, and the patients who lined up pretty immediately for enrollment in the trial.
Certainly can understand that. Well, thank you, Dr. Schendelman, for such a wonderful review of the program. And you mentioned that it might be you're going to be fully enrolled in your study in the near term, and yet it's a 2-year study. So I'm guessing some of the conversations with the FDA about looking at a biomarker and potentially some functional readouts that you can do along the way to see how the trial is going. And that's presumably what you want to do versus waiting 2 years for the big readout.
Yeah, you're exactly right, Tom. So we're looking at sorbitol reduction at 3 months, and I think it's been a very productive and active dialogue, amongst many members of the community with regard to the urgency to treat many of these diseases, and use of some of these accelerated approval pathways that are potential way forward in the US from a regulatory perspective. So we're hopeful that, the favorable safety profile that we have, because we have been studying this drug for quite a long time in another rare metabolic disease. So we have a phase 3 trial ongoing in another disease called galactosemia, which also involves treatment of children, starting at the age of 2. So we're pretty hopeful that a favorable safety profile and the urgency here, will allow something like accelerated approval. But you're absolutely right.
Well, thank you. And it's good to hear from you and David as well that your partnership with the FDA, and of course, having a close partnership with the agency will only be very helpful as you continue to advance your drugs. So let's then turn to Dr. Prasad. All of us are very excited conceptually around gene therapy, and there are now approved drugs that utilize gene therapy. And what's exciting about that, at least I will say, has curative intent. And so in the last year, Dr. Prasad, as I kind of noted at the top, it's been a tough year generally in the biotech community in terms of funding for companies and the overall economy. But if you can just let the audience know what Taysha's been up to the last year that either directly or indirectly impacts CMT, and what the company is considering around CMT and addressing particular subtypes of the disease.
Absolutely. First of all, Tom and the rest of the organization, thank you for the invitation to come and speak and share some thoughts on the panel. Yes, my name is Suyash Prasad. I'm a pediatric intensivist by training and the Chief Medical Officer, Head of Research and Development at Taysha Gene Therapies. We're based in Dallas, and our approach has been to develop AAV9 gene therapies for the treatment of neurological disease. And our technology is based on the work of Stephen Gray, who is a very eminent gene therapy researcher based out of UTSW in Dallas. And we have a whole host of programs. We have many programs in the realm of neurological disease, mainly pediatric and mainly rare disease. I'm pleased to acknowledge, Tom, that it has been a tough year for the biotech environment. Financially, it's been the biggest downturn and the longest downturn in the history of biotechnology financing, and that has unfortunately led to some, I guess, has led to a situation where funds are not as readily available to move programs forward as rapidly as we would like. Accordingly, we've had to slow down some of our programs at Taysha, and we are focusing on 2 lead programs currently. One of them is for Rett syndrome, which is a neurodevelopmental disease that many of you will be familiar with. The other is much more relevant to the discussion today, which is an AAV9 gene therapy approach towards treating giant axonal neuropathy, which is an axonal neuropathy that has many commonalities with CMT type 2. And we're doing this in partnership with our friends and colleagues at the National Institutes of Health, Carsten Bonneman, who's the principal investigator, and Diana Garusha-Gabal, who I think you know well, and I think may have spoken on a panel earlier today about the giant axonal neuropathy trial. Thus far, 14 patients have been dosed with gene therapy. AAV9 is the capsid. This is the viral shell that holds the DNA that codes for the protein that's missing. The protein that is missing in giant axonal neuropathy is gigaxinin. And 14 patients have been dosed intrathecally. So we give the drug, it's about 10 to 12 mils of drug is given into the spinal canal through an injection in the back through the spine. First patient was dosed a number of years ago, and we're at the point now where we are seeing in these 14 patients that have been dosed, of the 14 patients, 12 of them have received the therapeutic dose. It was a dose finding study. 12 have received the therapeutic dose with with highly relevant clinical benefit. And so the next step is to talk to the FDA and also the European regulators shortly thereafter to map out a pathway to an approval for this gene therapy medication. So that's where we're up to at the moment. We have 14 patients dosed. We've made enough gene therapy drug to dose a significant number of additional patients. So it's about another 50 patients worth of drug has been made, ready to go. We're working on all the relevant CMC. CMC stands for chemistry, manufacturing, and controls. We work on all the manufacturing type assays to ensure we can file a BLA for this drug. And we anticipate speaking with the FDA before the end of the year about how much more data they need to see in order for us to file for an approval for this medication. And when we find that out, we'll be able to enroll another 3, 5, 6 patients. It depends how many patients the FDA would like to see, move forward with that, and then file the biological license application. And then shortly after our engagement with the FDA, we will also be talking with the EMA, the European Medicines Authority. Let me stop there, and I can talk for many, many more minutes. Let me stop there and see if there's anywhere you'd like to focus on.
Well, so first of all, congratulations to your developmental, success, for Rett syndrome, and it sounds like you're well on the path to having an approved therapy. I think it would be probably really helpful because I'm guessing that you have now will be in a platform, meaning your AAV9 vector that you would use for CMT. And clearly would probably use the same route of administration as well, which I think would be so. Really, the obvious question here, Yash, is that I think it would seem to me, and please let me know if I'm overstating this, that everything that you're learning and moving forward with your application for Rett syndrome will be directly applicable for various CMT types that could be of interest to the company.
Sure, but perhaps maybe maybe the signal dropped out, Tom. But we do have one program for Rett syndrome. But the program I'm spending most time talking about was giant axonal neuropathy, or GAN,
Right,
which has a lot of parallels
Okay,
with CMT type 2. So
sorry, my mistake.
that's all right. Yeah, but yes, we're moving forward very nicely with this axonal neuropathy, giant axonal neuropathy filing approach with the FDA. And you're right. I think there's a lot of learnings from this program because we have a platform approach. Everything we do is AV9. Everything we do is given intrathecally. All the gene therapies we make are in the same. It's a mammalian cell system that's very well understood. So there are many platform learnings for this giant axonal neuropathy approach that can be translated and used for other forms of CMT. Absolutely, as long as we know what the mutation is, and as long as we know what the protein that's missing is, and as long as we can package the DNA that codes for the protein in a capsid, this approach is absolutely translatable to other forms of CMT.
Is the company giving any guidance when you will be, officially involved again in CMT and initiating clinical studies?
At the moment, no. At the moment, our programs are giant axonal neuropathy and Rett syndrome. Those are the two we're focusing on. We do have programs in CMT at an earlier stage. I think, to be frank, once the biotechnology funding environment improves, we will be able to reinvest in some of our earlier stage programs. So it remains to be seen, but we have many programs for a number of neurological diseases that are on pause at the moment. When the funding situation improves, we'll absolutely reinvest in those.
Well, what's so exciting about UT Southwestern, of course, is, as you know well, it's such a center of excellence in working with Dr. Gray, and your partnership with Taysha really is very promising for the field. And I think what's really clear today compared to last year, we're talking with 3 companies, all of whom are in late stage development for drugs targeting what are reasonably rare diseases, but are advancing those just based on early signs of clinical success and really targeted therapies. And something we're not talking about today is that, in fact, there are additional companies as well that have earlier stage programs, but will be entering the clinic in this year and in early next year as well. And of course, we all hope and we kind of live in this biotech environment, which is feast and famine, that goes up and down. And of course, it's all cyclical, and it will return to better days. And part of the reason is that companies like each of yours are really what generate this next generation of drugs that ultimately partner with large pharma. And it's really exciting to see. I think with that, we're just about out of time. But I do want to give an opportunity for each of you to say anything if there was something that was missed that you want to point out, because at least speaking for myself, I'm really very excited about the advances that each of you have made in the past year. I'll start with you, Dr. Solomon. Do you have any last comments you'd like to make?
No, we're just delighted to be able to work towards improve the health of patients with CMT1A in our case, their families and physicians. And I think what's exciting here is that we're close to having the first medicine for CMT1A, having data. So we like to promise less and deliver more. We're excited for the potential, and we look forward to sharing any results we may have with the community in the next year and a bit.
Great, thank you. And Dr. Schendelman?
Thanks, Tom. I'll plug the next session if that's okay, but I think there are breakouts sort of immediately following this on different subtypes of CMT. So our Chief Medical Officer, Dr. Ricardo Perfetti, is speaking in more detail about SWORD in a breakout session. So if anyone's interested in more information there, please watch the next session. And just another big thanks to the CMTA, to the researchers who really, advanced SWORD and have partnered with us, and most importantly to the patients who, just have been above and beyond what we could expect in the clinical trial. So thank you, everyone.
Great. Okay, well, with that, I think we need to wrap up. And I will just say thanks to each of you and to the audience listening in, and really can't wait until next year to hear all the new developments and the hope for success that we all share. Thank you.
Thanks, Tom.
Thank you.
Bye.
Thank you.
Thanks, Tom. This is Jill here. I'm here with Catherine, and we're just going to take a couple of minutes of transition and sharing information before we go into the individual breakouts. Obviously, thanks to all the panelists and all the doctors and the partners. This was an amazing set of sessions. I think, as I was hoping when I introduced it, I think it gives you a taste, at least, of the breadth and the depth of the STAR program. And again, this is just the tip of the iceberg. We have 40 partners, 50 projects, and we're starting to see some of those projects, as you saw right now, that are hitting the clinic and are getting to patients, which was just a dream a few years ago. This being said, we're not done yet. I mean, I want everybody to understand this is just the beginning. Drug development is a long, expensive, risky, tough, disappointing process. A lot of things end up not working, but the fact that we have so many partners and so many projects is the most important signal that a lot of things will work and we'll learn from whatever happens. So there's this really snowballing effect happening right now, which really was our vision a few years ago, and now it's happening and gives us a lot of optimism. But we got to be patient, and this is not the time to let the foot off the pedal. We got to also pedal the metal and continue to push. So to that respect, Catherine and I are going to say a few words about this, because as I said from the beginning, this is your program. I mean, every dollar that goes into this, everything you've heard about was funded by the community. So you should be very proud, okay? And we got to continue. But you've heard from a lot of companies, the reason why they came in is because we had the right animal models, the right testing, the right biomarkers. All this was done by the community, done by you, okay? So we got to continue. So keep that in mind. And I kind of want to relate back to something that our first speaker said, Kenneth. He said a lot of cool things, but he had one quote. He said something like, I really wanted a better life, but I didn't know how to ask. Remember that? And I tell you, that's the way it felt for us as a family 10 years ago, or 20 years ago, actually, when Johan was diagnosed. We wanted to help. We wanted to do something. We didn't know what to do. And there wasn't much you could do. Well, today is very different. There's a lot that every one of us can do. And we can all make a better life for us and for people with CMT. And from the STAR point of view, obviously, we can help fund the program, but also more and more, we need patients. That's why we created this program called Patients as Partners. It's incredibly important because all the companies you heard about, they want to talk to patients. They want to understand you. They want to read. It's not just for clinical trials. It's for the whole understanding of the disease and the patient and the people behind it. And the more they move to clinical trials, the more they need patients. So please, and Catherine will tell you how, join the patient and partner programs. And then you heard from Laurel and also from Jonah, or all the other activities we have as well. So Catherine will summarize all this for you, because now there's a lot we all can do, and let's just do it, okay? Thanks. You're on mute. Catherine, you're on mute. Catherine.
So sorry. First time I've done that today. Always a good time for it. So if you have been listening to this afternoon sessions and you've been thinking, what can I do to help? Well, then I can tell you how. So all of the experiments and all of the trials that you've heard about this afternoon would just simply not be possible without the CMT community. This research that we're doing, it's for you, but what we really need is your help to make it happen. So we can't build a pathway to clinical trials without the CMT patient community. We're asking you to raise your hands to participate in CMT studies like the ones you've heard about today. And lots of you have been asking in the chat, how can I get involved? So natural history studies, they're the ones where they want to know about your CMT, how it affects you and how it changes over time. Those natural history studies, they're not possible unless you consent to your anonymous clinic assessment data being added to the big national database of CMT patients. We don't know what you might be looking for from a clinical trial unless you come along to one of our patient-friendly focus groups and tell us about it, and the companies who are running the trials, they want to hear from you. It can be just as simple as filling out a questionnaire. It can be logging your symptoms on the CMT and Me app, or it could be popping on a wearable sensor that monitors your movement while you go about your day. Some of us might be lucky enough to be able to provide a blood sample for genetic analysis, or even donate some stem cells. Any way that we participate, it benefits every person who's got CMT, and it helps scientists understand our disease and develop treatments for us. And what we especially need is more people in their 30s and 40s to enroll in studies. It's not an enormous commitment, and there's often a small stipend to help with travel expenses for studies that you might need to attend in person. If you want to get involved, then all you need to do is visit the CMTA website. One of my colleagues will post a link in the chat. You go to the Patients as Partners page and you click to create a profile. And then you just fill in as much information as you can about your CMT. I built a Patient as Partners profile for myself, and now I get regular updates on my CMT type, what's going on with it, and what opportunities there are. Don't worry if you don't know your type of CMT yet. You can still get involved. You'll also then be the first person to hear about new treatments, and if you meet the criteria, then you get an early opportunity to access parts of clinical trials. I feel really strongly that this current generation of CMT patients, us here right now, we are blazing a trail. We are lighting the way on our path to developing those new treatments for CMT. And beyond joining Patients as Partners, there are also some other ways that you can help advance CMT research. Because there's too many of those to chat about, I'm just going to bring up one slide that's got a bit of information on it. So there's lots of different things you can do, and I'm sure Gilles might jump in on some of these too, because he's involved in them as well. One of the things that you can do is to just help the CMTA raise awareness. If people don't know what CMT is, they're less likely to want to run studies on it or get involved in any research. So what you can do is share your story. You can do that through friends, through family, locally, nationally, internationally, tell the whole world. You can do that through social media, and the CMTA can help you do it. Just last month, the CMTA shared my story about a trip that I took with my 8-year-old son to have a hip x-ray at our local hospital. They were just checking to see if he had hip dysplasia, which can be more common with people with CMT. The CMTA team made it really easy for me to share that story, and hopefully that provides some support to other parents and reassurance to young children who might be going in for that hip x-ray next time. The CMTA would really love to hear your stories too. As Gilles said, we can't do any of the research that you've heard about today without fundraising, without donations from the CMT community. And that be walking, cycling, dancing, streaming, or you can just own it and raise funds to support CMT in whatever way that you enjoy. This handsome devil right here, the man in the yellow lycra, that's my husband. He's called Ben. And last month, he was only just about standing up in our back garden after he completed his first ever 100-mile cycle to raise funds for CMT research. He and a few of his colleagues from work, they got together, they trained really hard, they cycled 100 miles, and they've raised over $1,000 for the CMTA. If every family with CMT could find a way to raise this amount of money by whichever way they want to, we would have millions more dollars to accelerate research. The CMTA is also all about connection. So through joining the CMTA, and volunteering at Camp Footprint, for the first time, I found my tribe. I joined the tribe, and I can now be surrounded by people who just understand without me having to explain. You can find your tribe by getting involved in your local CMTA branch, maybe through the Compass group, or connecting with the CMTA youth, who are just the most incredible group of young people with CMT. You can also send your children to Camp Footprint. I'll be sending my son when he's old enough to go. If you're too old to be a camper, you can volunteer to be a camp counsellor. If you can't get to camp, then there are lots of other opportunities through the CMTA's network in your local state to volunteer. All of the details of our face-to-face events and volunteer opportunities are on the CMTA website, so I hope you'll explore it and find a way to get involved. Because this is a virtual summit and we've seen little squares of each other all day, what I'd really hope and encourage you could do is you can connect virtually with me, the CMTA staff, and the CMT researchers. You can sign up for our e-newsletters, but you can connect with us on Facebook, Instagram, Twitter, TikTok, LinkedIn, whatever your social media poison, the CMTA is on that platform. And even if you love podcasts, we've got one of those as well. So my just final reminder and plea is just to say, please build a profile on Patients as Partners in Research and join in. It's really just as simple as filling in a form. And then what we really hope is that we'll get to see you all in person before next year's summit. But just stay tuned for announcements about 2023 conference dates, and we're going to have face-to-face and virtual options. So thank you again to all of the panel experts who've joined us for this afternoon. It's shortly time for us all to go into the subtype-specific breakouts. In those breakouts, you'll be able to use the Q&A function, just as you did this morning, and you'll get a notification from Emmy, head on into that lounge and look at the agenda, and you'll be able to pick your subtype room and go over there and listen to some more information on your subtypes. So thank you for joining us this afternoon, and we'll see you in the subtype breakouts.